Hi, I'm Ferdous Nasri, a scientific researcher and bioinformatician at the Hasso-Plattner Institute. Here I'm presenting the poster on pathogenic potential prediction of novel fungal DNA using ResNets based on a newly curated fungi-host database. Fungal disease outbreaks kill hundreds of thousands every year, and they're hard to predict and diagnose. Pathogenic fungi are also under-researched while they're constantly evolving to survive in new environments and hosts. At the same time, sequencing technologies have advanced rapidly, opening the door for computational approaches to predict the pathogenic potential of fungal species. In order to train machine learning classifiers, we first need a large amount of labeled data. So the first step is to curate a database of fungal species along with their labels on pathogenicity towards humans, plants, and non-human animals. For this, we used many different resources, the top three being the U.S. National Fungus Collection from the USDA, and the Atlas of Clinical Fungi, and a paper from Taylor et al., which labeled many pathogenic fungal species. We ended up with a manually curated database of 982 labeled pathogenic fungi. We further analyzed the 471 species with genomic data and decided to define a binary classification for our machine learning classifier. Research suggests that fungal species which can be hosted on or in humans could act as opportunistic fungi given the required conditions. For example, in an immunocompromised person. For this reason, we chose our positive class to be the human-hosted fungi and the negative class to include plant and non-human animal pathogenic fungi. On the two horizontal bar charts on the left, you see how few of these species have a complete genome sequence available. On the right, you can see a UMAP plot of the learned genome representations. This plot shows how the two classes are nicely separable from one another. Blue dots representing the negative class and red dots representing the positive class. We trained a modified ResNet classifier which uses one-hot encoded sequences as input. For this, we use the approach described in the paper on deep learning-based real-time detection of novel pathogens during sequencing. And we compare our results with that of the state-of-the-art homology-based classifier BLAST. As you can see in this table, we outperform BLAST in accuracy on all three test runs, with single 250 base pair reads, which you would get from a MySeq Illumina machine, read pairs, and also full genomes. Note that BLAST does not always provide a classification, and therefore the prediction rate at the bottom of the table are sometimes less than 100%. Finally, we look at the performance of a multi-class classifier. For this, we take viral and bacterial datasets from published work on machine learning trained predictors for such a task. On the first table here, you can see the performance of different bacterial and viral classifiers compared with BLAST. On the left for bacteria, the two-class classifier differentiates between bacterial pathogens and non-pathogens. The three-class adds the viral pathogens as another dataset, and the four-class adds the human-hosted fungi. Same is done for viruses. As bacteriophages and the pathogenic bacteria they infect seem to have high sequence similarity, we reassigned the bacterial pathogen class to the non-pathogen class and mark that with a reassigned on the table to be able to capture this effect. We can see that multi-class ResNets outperform BLAST in accuracy every time. On the table below, we can more closely observe the effect of adding fungi datasets to the others. We observe that the human-hosted fungi are easily differentiated from the other pathogens as they do not have a large negative effect on the predictors. We thank you for your attention. If you have any questions, do not hesitate to ask us here or per email. Bye!